Good evening, sir, and all my batchmates. My topic is different types of vaccines. Vaccines denote all biological preparations produced from the living organism that enhance the immunity against the diseases. Vaccines either prophylactic and therapeutic. Vaccines are administered in liquid form, either by injection, by oral or intranasal route. Types of vaccines in brief. Live attenuated vaccines, measles, mumps, and chickenpox. Inactivated vaccines, hepatitis and influenza. Subunit vaccines, hepatitis B. Toxoid vaccines, diphtheria and tetanus. Conjugated vaccines, HIV. DNA vaccines, vaccines against influenza and herpes. Recombinant vector vaccines, vaccines against HIV and rabies and measles. Live attenuated vaccines inject into the body into live version of the pathogens or virus that causes the diseases. These type of vaccines are most similar to those actual diseases. They are more effective and often only require 1 or 2 doses of the lifetime immunity. Live attenuated vaccines should not be given in individual and weak and damage immune system. To maintain potency of the live attenuated vaccines, the refrigerator protect from lights. Next, sir. Hepatitis and influenza. Next slide. Subunit vaccines. Only the portions of the germs that cause an immune response are used to create a subunit vaccine. These portion of the germs are called antigens. Subunit vaccines can contain from 1 to 20 antigens, since only the specific necessary part of the germs are used to these types of vaccines. Following methods are identified and those obtain the antigens, grow germs and pack in part from the harvest necessary antigens, create antigens molecules using DNA technology, hepatitis B vaccines. Next. So एक slide इसमें रह गई है inactivated vaccines. Second. Conjugated vaccines. Conjugated vaccines are the HIV and pneumococcal conjugated vaccines. Similar to subunit vaccines, conjugated vaccines are only portion of the germs. Many bacteria molecules are coated by sugar-coated polysaccharides. This coating hides and diffuse germs antigens that immunate immune system for infants are not able to recognize it. Therefore, the scientists attach to polysaccharides stronger portions. Immune system responds to the proteins, it also responds to the polysaccharides. DNA vaccines. DNA vaccines are synthesized based on the information of the DNA microorganisms. DNA vaccines are developed using the information by the diseases that code of those all important antigens. There are currently studies for DNA vaccines for herpes and influenza. Recombinant vaccines. Recombinant vaccines are similar to DNA vaccines, except the weak and attenuated germs is used to carry DNA to the cells of the stimulate immunity. Similar to the attenuated vaccines, recombinant vector vaccines are similar to the actual diseases. Vaccines against HIV, rabies, and measles. Thanks.